This is part 2 of the Chapter 16 recordings, and we're starting with slide number 43. What we're covering right now is the process of DNA replication, which occurs during the S stage of interphase, right? DNA replicates before the cell divides. And DNA replication is referring to the process where you have a single-stranded chromosome and it becomes a double-stranded chromosome with two sister chromatids. So, when we learn about DNA replication, we are going to learn about enzymes because there are enzymes involved in each little process, that occurs. DNA replication just means that you take one original DNA strand and it copies itself. So that's going to begin with an enzyme called helicase. Helicase, because remember the DNA is a double helix, so an enzyme is going to be named, an enzyme is going to be named for what it does. So helicase is named because DNA is a double helix, and helicases are enzymes that untwist the double helix. They unwind and they separate the two strands of DNA. All right. And then to keep it from binding, basically reconnecting, to keep those strands separated and not connecting back together, there are these proteins called single-stranded binding proteins. And they bind to and stabilize the DNA so that it will remain open. Then there is an enzyme called topoisomerase, and it says it relieves the strain of twisting of the double helix by breaking, swiveling, and rejoining DNA strands. So I'm probably not going to highlight that one because I do want you to know the role of helicases and the role of the single-stranded binding proteins. So here you can see a picture. Basically, you've got an original double strand of DNA. DNA helicase is this green molecule, and it just moves along the DNA, and it unwinds and separates the two strands. Then you have these little gray oval-shaped single-strand binding proteins, and they prevent the two strands from binding back together. Then we're going to talk about a sequence of RNA, and RNA is just single-stranded nucleotides rather than DNA is a double-stranded molecule. So RNA is still composed of nucleotide bases, just like DNA, but it's like half of the DNA. It's like just a single strand, or basically if somebody came through and cut a ladder in half so that you only had one side and half of the steps, that's what DNA or what RNA looks like. So the way that replication begins is that a little tiny piece of RNA called an RNA primer binds to a point on the DNA. And then an enzyme comes in. This pink enzyme is called primase. And primase is going to bind to the DNA at the point where the RNA primer binded to the DNA. And that is where replication is going to begin. Okay, so back to the notes. There's an enzyme called DNA polymerase, and I don't know why it's not bold. I'm getting ready to make it bold, if I can. I don't know if I can. Anyway, that's an important word. That should be in bold. Yeah, I just don't know how to do it, and it's going to take too long. So make sure you know what DNA polymerase, know that is one of your vocabulary terms, and it's probably the most important enzyme in the list of enzymes. DNA polymerase, remember that DNA is a polymer. It's a long chain of nucleotides. Polymers are long chains, okay? So polymerase adds nucleotides to the old strand so that the new strand is synthesized. So it basically synthesizes the new DNA strand. So before DNA polymerase can come in and add new nucleotides to the old strand, the primer has to bind. Okay, so the initial nucleotide chain is a short RNA primer, which I just showed you that. That is like a very short sequence of RNA that binds to the DNA. And the enzyme primase catalyzes that reaction, the binding of the RNA primer to the old strand of DNA. Okay. So then the new DNA strand will begin, and DNA polymerase is the enzyme that will add nucleotides to the old strand to make a new strand. Okay, now it's bold. So enzymes called DNA polymerases catalyze the synthesis of new DNA at the replication fork, which is just the place where the old DNA strand opened up. All right. Most DNA polymerases require an RNA primer and a DNA template strand. That's just a word for the old DNA strand. And there's a lot in here you don't need to know, so I'm going to skip it. But this is what it's going to look like. Okay, here's your old DNA template strand. And then what DNA polymerase is going to do is going to come in and add these nucleotides on the left side. It's going to add them in what's called the 3' to 5' direction. So it's going to, this is the old strand, the 3' end and the 5' end. I really don't care if you understand what that means. It has to do with the chemistry of the, they number the carbon atoms in this sugar molecule, and it's the 3rd carbon atom. So starting with the 3rd carbon atom, that names one end of the molecule. It's called 3' because it's named after the 3rd carbon in the sugar. And then this end is named for the 5th carbon in the sugar. And you can't see the sugar, but this line would be chemically bound to another sugar molecule. So anyway, basically, the 3' to 5' is just, we're just naming the ends of the DNA. And so if this is the 3' end, this is where our DNA polymerase starts adding nucleotides. And it knows to add A because A is complementary to T. It knows to add C because C is complementary to G. And then it adds G, it adds T, you know, using the rule of complementary base pairing. So DNA polymerase can add nucleotides only to the 3' end of a growing strand. So it's really adding, okay. The way they're wording this, it's adding in the 5' to 3' direction. I guess it's talking about this new strand is the 5' to 3' direction. But you can see the ones that come in are adding here. You know, they're adding to the 3' end of the new strand, basically. Sometimes it's really tricky how that's worded. But anyway, the DNA polymerases have a special direction that they can add nucleotides in. And so they can only add to the 3' end of a growing strand, and DNA can only elongate in the 5' to 3' direction. So one of the new strands is going to be synthesized continuously, and we call that strand the leading strand. Okay? So DNA polymerase synthesizes a leading strand continuously in one direction without stopping. But the other strand is not running in the 5' to 3' direction. It's running in the opposite direction. So it's called the lagging strand. You can see that in this picture. Let me show you. So when that DNA opens up, when DNA helicase comes in and opens up the DNA, it makes basically a replication bubble. And each end is called the replication fork. But one of the strands, which we call the leading strand, is going to be synthesized. If this is the origin of replication, this is the leading strand. It's going to be synthesized in the 5' to 3' direction. So from this end to that end, from here to here. And the lagging strand is running in the opposite direction, 3' to 5'. You see where its 3' end is here and its 5' end is here. So because it's running in the wrong direction, DNA polymerase can only add a few nucleotides at a time. So it does one little piece and one little piece and one little piece in little fragments. And they're actually called, they have a name, they're called Okazaki fragments. So then for the lagging strand that's only been replicated in fragments, another enzyme has to come in called ligase and link those fragments together. So there's a leading strand that's replicated continuously. Then there's a lagging strand that's replicated in fragments, and it has to be sealed together by the enzyme called ligase. So here's an animation in slide 52 that would be good for you to watch. This animation only shows you synthesis of the leading strand, which is the one that DNA polymerase replicates continuously. But for the lagging strand, DNA polymerase is working in the wrong direction. So it synthesizes that lagging strand in segments called Okazaki fragments. And those fragments are then joined together by an enzyme called DNA ligase. And remember, that only happens on the lagging strand. The leading strand doesn't need DNA ligase because it's replicated in one long strand instead of in segments. Okay, so this is just showing you that the Okazaki fragments that are produced, and then DNA ligase coming in. DNA ligase is this pretty shade of aqua, this pretty aqua shade, but it's coming in and it's linking the Okazaki fragments together for the lagging strand. And here's an animation in slide 55 on the synthesis of the lagging strand. So basically, then this is a good one because it puts everything together. DNA replication, and it shows you the synthesis of the leading and the lagging strand. So this would be slide 57. All right, so here are the enzymes involved, and I'm not really going to get to, this is really the replication of bacterial DNA, so I'm not going to get too caught up in DNA polymerase 1 and DNA polymerase 3. I just want you to know helicase unwinds and unzips the DNA, the parental strand of DNA. The single-stranded binding proteins hold those strands open so they don't seal back together. They don't come back together. Primase is the enzyme that causes the RNA primer to bind to the DNA, and that has to happen before DNA polymerase can add nucleotides to synthesize the new strand. So DNA polymerase comes in and synthesizes one new strand called the leading strand, and it synthesizes the leading strand continuously. And then on the other strand that's running in the opposite direction, it's running in the 3' to 5' direction, the DNA polymerase synthesizes the new DNA in segments, and they're called Okazaki fragments. And that strand is called the lagging strand. And once those Okazaki fragments have been synthesized, they're joined by DNA ligase. So that's kind of a summary of DNA replication. All right, now the really good thing is that DNA, when DNA replicates, it does make mistakes. So another thing DNA polymerase can do is it can proofread and it can check for mistakes. And mistakes being like instead of T pairing with A like it's supposed to, maybe an A pairs with an A, or a G pairs with an A. So DNA polymerase proofreads the new DNA strand and fixes errors, replaces any incorrect nucleotides. And that is called mismatch repair. No, it's not. I'm sorry. First, there's proofreading. And if proofreading doesn't work, then there is mismatch repair. And there are other enzymes called repair enzymes that have evaded the proofreading process. I'm sorry, I made that more complicated than it needed to be. So the first thing that happens is DNA polymerase proofreads, and it fixes some mistakes. Then in mismatch repair, repair enzymes fix mistakes that DNA polymerase didn't fix. One of the repair enzymes is called nuclease, and there are different ones. And I'm going to skip through some of this just because we don't have time to cover everything. Okay, there is a section on the ends of DNA are called telomeres. So our chromosomes have telomeres, which are just special nucleotide sequences at the ends of our chromosomes, okay? And those telomeres shorten as we get older. And that's one sign of the aging process. So it's been proposed that telomeres shorten, the ends of our chromosomes shorten as we get older. Okay, so there is an enzyme called telomerase, which helps to lengthen the telomeres and prevent aging. Okay, in the eukaryotic cell, this is kind of a review, but in our cells, which are eukaryotic cells, our DNA is in a form called chromatin during interphase, and then whenever it divides, it condenses into chromosomes. So chromatin consists of proteins called histones, and the DNA is wound around these proteins called histones. And when DNA is wound around the histones, we call that a nucleosome. And so basically this shows you that during interphase, or during prophase, the chromatin, which consists of DNA wound around histone proteins, starts condensing. And the first stage that it forms is called euchromatin. And then the second stage where it's really, really super condensed and we can see these chromosome structures that look like an X, the letter X, your two sister chromatids, you can see them clearly under the microscope. That's called heterochromatin. And that's what we see by the end of prophase. We see heterochromatin, and that is the name for the condensed chromosomes that we see, during prophase and during mitosis. So loosely packed chromatin is referred to as euchromatin, and then condensed chromatin is referred to as heterochromatin. So when we say that chromosomes condense, or chromatin condenses and forms chromosomes during prophase, that form of DNA is called heterochromatin. That is the condensed chromatin that you see during mitosis. And this is just showing you, like you can take the percent of each, if you know that a sequence of DNA has 32%, we're just going to round these, or just go back to the whole number. Let's say the DNA is 32% adenine. Well, it's got the same amount of thymine, 32% thymine. Then what you do to figure out the guanine and the cytosine is you add 32 plus 32, which is 64. And then 64 from 100 is 36. So you're going to get 36 divided by 2 is about 18. So you're going to have about 18% guanine and cytosine. And I know it's not quite 18% because of our decimals everywhere. But you end up with the same percentages of guanine and cytosine, and then the same percentages of adenine and thymine. And if you know the percentage of one, like down here, if we know that an ox has 29% adenine, then it also has 29% thymine. Maybe I can do this. I'm going to try. Maybe I can draw. We're going to do a little experiment. All right, so I'm going to use red. Okay. So if an ox has 29% adenine, then it also has 29% thymine. So far, so good. All right, 29 plus 29 is 58, right? 58, yes. All right, what is 100 take away 58? That is 42. So guanine and cytosine has to add up to 42, which means they're going to be 21% each. So hopefully you got that and understand how to do that. So now we just add up and make sure we get 100. 29 plus 29 is 58. 21 plus 21 is 42. And 58 and 42 equals 100. All right, so I don't know how to go back to the pointer, but anyway, I think we're about done. So again, when we look at a DNA molecule, the sides of the ladder are composed of sugars and phosphates. So each side of that ladder is a sugar-phosphate backbone, and the steps or the rungs of the ladder are the base pairs, the nitrogenous base pairs. Just to review a little bit. All right, that's it. Okay, so those are the recordings for chapter 16. And now, hopefully, if you have any questions, you can ask and get your assignments done for chapter 16 this week.